All right. Well, good morning, everyone. I think we'll go ahead and get started. So thank you so much for joining us bright and early this morning at this joint presentation between the Michael J. Fox Foundation and Abcam, where we will be presenting to you on the biomarkers of neuroinflammation and Parkinson's disease. My name is Elnaz Atabakhsh, and I am the Senior Product Manager for Multiplex Assays at Abcam, and I will be one of your presenters this morning. I'm going to be joined by Dr. Nicole Polinski from the Michael J. Fox Foundation. She is the Associate Director of Research Programs at MJFF. And for today's talk, we're going to begin by telling you a little bit about the introduction into Parkinson's disease, as well as the foundation itself, and the importance of investigating inflammation in Parkinson's disease, which will be the focus of today's seminar. After that, I will join you where I will take you through the FirePlex technology platform, which was used to do all of the biomarker profiling studies that we'll be talking about today. And then Nicole and I will present some summaries and future directions for you. So with that, I'm going to hand it over to Nicole to get us started.
Great. Thanks, Elnaz. So I first want to start, for those who may not be as familiar with Parkinson's, with just a brief overview of this disease and what we currently have in terms of understanding as well as therapeutics. Parkinson's was first described by Dr. James Parkinson in 1817 on his essay on the shaking palsy. It is the second most common neurodegenerative disease after Alzheimer's. Approximately 1 million individuals in the United States have Parkinson's, and the global incidence is approximately 6 million individuals. Importantly, the prevalence of Parkinson's is projected to double by the year 2042, so it's very important that we start to uncover more about this disease, how to treat it, and how to get patients better care. The symptoms for Parkinson's include motor and non-motor symptoms. The motor symptoms are the classical Parkinson's symptoms. It's what diagnosis is based off of. These include tremor, bradykinesia, reduced arm swing, balance and gait issues, amongst others. Non-motor symptoms can occur decades before motor symptoms show up, and this can include symptoms such as constipation, anosmia, sleep issues, and then in late stages of Parkinson's, cognitive issues as well. Importantly, only about 10% of cases have been linked to a genetic mutation, so we still have a lot to learn about the other 90% that are sporadic or idiopathic. And we do know some risk factors for Parkinson's disease, and these include advanced age, head injury, pesticide or heavy metal exposure, among others. This is probably pretty small, but just some key important takeaways is we don't actually have any disease-modifying therapies for Parkinson's disease currently available. The best Parkinson's drug was discovered in 1967. That's levodopa, and that was when Lyndon B. Johnson was president and Neil Armstrong had not yet walked on the moon. So although that's a really good drug for patients, it really helps them with their motor symptoms and some other symptoms. We have a long way to go, and we're really trying to make progress on that front. We at the Fox Foundation see Parkinson's disease as a complex puzzle. This includes various pieces that need to kind of come together to give us a holistic view of what's going on in this disease so we can help patients. Some of these pieces we know may include a genetic component. We're really invested in understanding the pathways underlying Parkinson's, like neuroinflammation. Diagnosis, early diagnosis, better markers for disease progression, and more accurate diagnosis are also going to be very important for this disease. And then clinical experience, both having patients involved in clinical trials and getting patients better treatments is very important. And then we have all of these other pieces that we have still yet to uncover and tease out to get a very good understanding of what's going on. At the Fox Foundation, we believe progress requires funding and field-wide coordination. So we work across various spectrums at the foundation. We have investment in patient cohorts, enrolling patients to provide either biosamples or participate in clinical trials, things along those lines, because it's really the patient involvement and cooperation with us that's going to move this dial forward. We also have initiatives in research tools. So we've found that it can cost a lot of money and a lot of time for researchers to make the tools they need for their experiments. And if we have tools ready-made for them, we can really move progress faster. We have a lot of work understanding the biology of Parkinson's disease. be that what's going on in idiopathic Parkinson's, in pathways that may be implicated, as well as in the genetic forms of Parkinson's disease and the risk factors we've identified. Biomarkers, which is kind of the key to this talk, will be very important for Parkinson's disease research. It'll help us not only diagnose the disease better, but maybe earlier so we can start interventions sooner. It can help us monitor disease progression, which may shorten clinical trials and kind of find the diamonds in the rough that we may be overlooking now. And then therapies. The foundation is very involved in getting better therapies for symptomatic motor symptoms, non-motor symptoms, and trying to uncover some disease-modifying therapies for patients as well. And across all of these different areas, we have crosstalk between the different groups, and we really find that accountability, collaboration, diverse expertise, open access resources, and validation and replication are so important, and it's what's really going to move the dial. So just a little bit of information on the foundation. The Michael J. Fox Foundation is the world's largest nonprofit funder of Parkinson's disease research, and our mission is to accelerate the development of improved therapies and ultimately a cure for Parkinson's disease today. And our ultimate goal is to go out of business because we've met that goal. Our stats, we are a public charity that was founded in the year 2000 by the actor Michael J. Fox. We have more than 93,000 donors last year and over $800 million in research programs funded to date. We have more than 700 active grants in our current portfolio, and we fund where the science takes us, with 1/3 of our funded projects with researchers outside of the United States. And we fund across the spectrum. We fund academia, industry, biotech companies, all groups that will help this understanding. Our research priorities fall into 3 different buckets. We're invested in looking at symptomatic treatments for motor and non-motor symptoms, as well as emerging targets. Disease modifying by looking at the genetic underpinnings of Parkinson's disease, proteins that we know go awry in this disease, and also emerging targets like inflammation. And field-wide challenges by addressing the need for better biomarkers, clinical trial recruitment, research tools, and we also have a small public policy and advocacy arm to voice patients' needs on Washington, D.C. We prioritize and pursue research in these areas by both providing financial and intellectual support for projects from discovery through the clinic to ensure that progress towards new therapies for Parkinson's is made. And for this talk, I'll really be focusing on inflammation, which falls especially under both the disease-modifying bucket as well as field-wide challenges such as biomarkers. So what is known between inflammation and Parkinson's that have linked these two things together? Well, first, inflammation's been linked to Parkinson's through genetics. LRRK2 and Parkin, which have been linked to an increased risk for Parkinson's disease, play a role in the immune response to things such as tuberculosis and leprosy. And mutations in immune mediators have been linked to certain cases of familial inherited Parkinson's disease. So through genetics, we're starting to understand that inflammation may play a role either on its own or in collaboration and synergy with some of these other mutations that patients may have. We've also linked inflammation to Parkinson's through epidemiology studies. There have been reports that chronic use of select anti-inflammatory medicines have been associated with a reduced risk for a secondary Parkinson's diagnosis. We've linked inflammation to Parkinson's through pathology studies as well. We see an increased presence of activated microglia and cytokines in post-mortem Parkinson's brains and CSF. And we see that inflammation is linked to Parkinson's disease through animal model studies as well, with various Parkinson's models having a neuroinflammatory component. And if you interfere in that inflammatory process, you can reduce degeneration as well. So we know inflammation may be related to Parkinson's, but why is it so important to investigate inflammation in PD? Well, first, as I said with the genetics portion before, there's a potential overlap with the usual suspects of Parkinson's disease, such as these mutations in LRRK2, Parkin, GBA, alpha-synuclein. And that leads to the question, does modulating inflammation affect PD-related targets or vice versa? So can we either impact these different targets and that may have a role on inflammation, or can we interfere with the inflammatory process going on and then have a better outcome for these mutation carriers? The immune system may be an environmental modifier of Parkinson's disease, maybe through epigenetics or cumulative risk. Maybe it's a multi-factor hit that leads to Parkinson's disease in the cases that are idiopathic, or the cases where patients have mutations and some go on to develop the disease, whereas others do not. There's a potential for a disease-modifying therapeutic development as well if we understand if inflammation is either causing Parkinson's to progress or contributing to the pathology as well. And for this, we can take a page from the MS playbook, hopefully, and kind of learn from what other fields are doing as well. And then, of course, inflammation is automatically biomarkerable. It's a secreted factor. There are markers on the outer cell membrane. You have peripheral readouts of inflammation in certain cases. So if we can understand the immune signature of Parkinson's disease, hopefully this will help us develop biomarkers that can help clinical trials choose patients for the right studies. Maybe we can use these biomarkers to help diagnose PD earlier and start interventions before pathology has increased to the point where motor symptoms develop. And as well, maybe we can use these biomarkers to see if a therapy is slowing or stopping or reversing disease progression. And this would be a really good readout for trials so they can be shorter, more focused, and we can have better readouts of efficacy. So the MJFF strategy for investigating the role of inflammation is threefold. First, we want to identify the PD immunophenotype, and we have key questions and challenges within this area. First, what is the PD-specific signature for both neuronal and peripheral inflammation? Are these different? Can you use peripheral inflammation as a readout of what's going on in the brain, or are they two separate components that need to be kept separate? Also, there's inherent variability and challenges for rigor and reproducibility with measures of inflammation. They can change and variate and fluctuate between times of day with circadian rhythms. They can change based on, if that patient's been exposed to the flu or things like that. So there are some certain issues and variability that we need to address in this field. So our strategy for looking at this is funding various labs to investigate inflammation using different assays and different readouts. We're convening an advisory committee to help guide us on the role of inflammation in Parkinson's disease and bringing non-Parkinson's inflammation experts into the fold to try and understand what other groups know and how we can move forward. We're looking at the role of chemokines versus cytokines. What's the impact of acute inflammation versus chronic inflammation in this disease? And should we focus more on one than the other? And finally, leveraging information, methods, and tools from other fields like the MS field to try and piggyback off on what is learned and what tools they have so we're not starting from scratch. Second, we're looking at inflammation biomarkers, and the key questions and challenges here are issues in translatability. If you're looking at animal models, the immune signature is very different than in patients in a lot of cases. Models are kept very isolated, microbiome is different, things like that. So really understanding how to move between species and how to understand what's going on and how you can interrogate these pathways. There's also a possibility that you may need to optimize new collection protocols based on what your readouts are. Are banked blood samples going to be the best if they're frozen, or do you need fresh collections, things like that? And we need to look at the possibility of developing companion biomarkers and integration of validation to see maybe how readouts of inflammation can be used with other readouts for Parkinson's to kind of get a more holistic look and a better feel for what's going on. And our strategy here is we launched a focused inflammation biomarkers RFA in 2017 as a call out to try and get groups to come to us with their ideas. We fund human biospecimen sample collections as well as cohort studies to try and bank samples to use in the future. And if we do need special collections, we will also do that as well. We prioritize companion biomarkers looking at inflammation in aging, phenoconversion, and microbiome studies, as these may all be very important in Parkinson's disease. And finally, we also have a lot of non-funding initiatives going on. So the questions and challenges we're addressing here are how can we leverage lessons learned and best practices from other disease areas, and how can that relate to Parkinson's disease as well? And then also developing quality imaging tracers and tools for inflammation that are lacking or not standard in the field right now. So our strategy here is to leverage knowledge sharing across disease areas to try and learn from each other. We're initiating consortia with other foundations to address broad field-wide challenges, such as the need for better tools or imaging markers. And we're generating new tools and curating information for animal models to help the community learn about this, what is known, and where to start when looking at this pathway. So our strategy is focused on implementing a strategic funding plan across targets, biomarkers, and therapeutic development by working with various groups to address these key questions and challenges. And important for today's talk is focusing on these two areas within identifying a PD immunophenotype and inflammation biomarkers, where we're working with Abcam to both, as funding their lab as one of our various labs to look at inflammation using different assays, as well as using our patient biospecimens that we've collected through our cohort studies to kind of move the dial forward and get the tools in the hands of the investigators. And with that, I will pass it over to Elnaz to go over our assay.
Thank you, Nicole. To begin with, I'm going to tell you a little bit about the FirePlex technology platform, which we use to do all of these biomarker studies, and then I'll take you through the experimental design and then the results. So the FirePlex technology platform was developed approximately 10 years ago at MIT, and it's based on the optical liquid stamping of hydrogel particles. And when this platform was created, it was specifically designed to be a multiplex platform for doing biomarker discovery and verification studies. And the platform as it stands now is designed to allow you to do multiplex detection of both microRNA and protein biomarkers directly from either tissues or biofluid samples without doing any sample processing or RNA isolation or anything like that up front. And the most important aspect of the platform is that it requires very small inputs of both biofluid and tissue samples. So it's very well designed for studies where you have limited samples and you're splitting it over multiple studies and you really need to get as much information out of it as you can. So the core of the FirePlex technology are these particles that you see here. And the particles themselves are these rod-shaped porous platforms that we use to do all of our analyte detection. The particles have 3 functional regions that are separated by 2 spacer regions, and I'm going to take you through the architecture of the particles just to explain how do we do our multiplexing and our analyte detection. So on either end of the particle, indicated in this slide in gray, we have the coding regions, and within these coding regions, we include different levels of yellow fluorescent dyes that allow us to do our barcoding and multiplex detection. In each batch of particles that we generate, we can look at up to 70 unique analytes because we can generate 70 unique code combinations. The center region of the particles, which is indicated here in red, is the analyte detection region. So for the microRNA platform, we conjugate DNA probes against the microRNA of interest. And for the immunoassay platform, we conjugate capture antibodies against the protein analyte of interest. Now, the particles themselves have several advantages over other bead-based assays based on the design and architecture that we use. The first is that they're made out of PEG, which is naturally porous. So rather than just getting capture on the surface like you do with other spherical beads, you actually get binding all the way throughout the particles. And this provides us with a very large capture surface for analyte detection. And so we routinely see about 5 logs of dynamic range in the assays that we develop. And the PEG itself, because it's biologically inert, you don't see any matrix effects with the other proteins and lipids that are present in biological samples. And so we can work directly with these sample types without having to do any sample processing. In addition, the 2 spacer regions that separate out the functional regions of the particles really do a great job of separating preventing interference and code bleed. So it's a really nice way of doing clean analyte detection. Finally, the assays are designed to be both multiplex and high throughput. So our standard assays are run in 96 well plate format, and we also offer assays that are run in 384 well plate format as well. So in each run, you can look at up to 70 unique analytes per well and up to 90 unique samples per plate. So you generate over 7,000 data points on each plate that you run, which is very important for the samples that we're looking at. The readout for the assays is done on standard benchtop flow cytometers, so it doesn't require any specific or fancy equipment, just a single laser and 3 detection channels. And then all of the data analysis is done with our integrated analysis workbench software. So to show you what that looks like, when the raw data comes off of the flow cytometer, we plug it into our software, of which I'm showing you a screenshot here. And the software does all of the upfront data analysis. So all of the particle decoding, analyte quantitation, statistical analysis, all of the case versus control comparisons, the patient groupings, all of that can be done within the software itself. The software is completely free and it runs on Java, so it can be run on as many computers as is needed. And all of the data analysis that I'm going to take you through was done using our software. So about 2 years ago, I approached our alliance manager, Sandy Chow, who was working with the Michael J. Fox Foundation, and I really wanted to learn more about some of the sample types that the Michael J. Fox Foundation uses for their biomarker studies to try and understand the challenges that are faced, particularly when you're working with things like CSF. And from that and a conversation with Nicole and other colleagues, it spiraled into a much larger project, which is why we're standing in front of you today. And so we set out to actually take on a proper investigative study to utilize the FirePlex technology platform to see if we could find biomarkers of neuroinflammation. And what we really wanted to try and do was to take as holistic and unbiased an approach as we could to looking at these biomarkers of neuroinflammation. So before I tell you about the study design, Nicole is going to come back to tell you a little bit about why it was important to try to take on this broad view.
Great. So we know inflammation and Parkinson's disease are probably linked in some way or another, but we still have some outstanding questions that we need to understand and cover before we can probably make some progress on this issue. So first, is inflammation a cause or a consequence of Parkinson's disease? Is it starting a cascade in the body that leads to pathology and degeneration? Or is it an artifact of that process that's already been going on and a readout of that? Or is it both? Is there an interplay between those two aspects? Also, what is the role of central versus peripheral inflammation? There's a lot of studies going on right now with the microbiome in the gut and how that may relate to Parkinson's disease pathology. We see a possibility of synuclein aggregation in the gut, and maybe that's an early readout of future Parkinson's disease. So is there something that's going on peripherally, maybe through the gut, that's causing an upregulation of cytokines, of these T cells, macrophages, that are leading to central nervous system inflammation? Or is it something that is coming from the top down as well, like aging in the brain, genetic mutations, or dysregulation of gene expression in the brain that's driving pathology the other way? Or is it a synergistic effect between these two? So another question is, should we be looking at chronic versus acute inflammation markers? Parkinson's disease is a long duration disease. It's thought that pathology may start decades before motor symptoms show up and diagnosis is made. So should we be looking at things that may be occurring over time and more drawn out, maybe chronic inflammation rather than acute inflammation that's important for other disease spaces? Or should we be looking at both? So what's the role of chemokines versus cytokines in this disease? So if we can tease that out, hopefully we can have a better kind of readout or inflammation profile for Parkinson's. In addition, previous work on neuroinflammation in Parkinson's disease to try to develop a biomarker has indicated that there are some other challenges that we need to kind of work through as well. So there are discrepancies in the published literature. Most studies report differences between healthy control and Parkinson's disease in inflammation readouts. But there are disagreements in the direction of this change. Some studies report an increase with Parkinson's, some report a decrease, some report no change. Some examples of this are interferon gamma and serum TNF as well. There have been multiple studies on these, but there's no consensus on what's changing in Parkinson's disease for these readouts. And this may be due to differences in disease severity of the patients that have been sampled, other comorbidities that are present, different protocols, individual variability, differences in sample processing, analytical methodologies, differences in sampling times, coupled with diurnal fluctuations of inflammatory proteins. The list goes on and on. So it's important to understand that all of these factors may be leading to the readouts that we have. So if we can tease this out, maybe by having very robust, stringent sampling protocols, standardized collection methods and storage methods, maybe that can help us kind of tease this out a little further. And then this is just a figure of a couple analytes that either vary across the time of day or across PD versus healthy control. So you can see that in kind of the tan color, these differ between PD and healthy control with little variability over time. In the middle differs both with time and disease state. And then in blue, these vary across time, but not necessarily PD and healthy control. So some of these changes may be masking any of the effects that we may be able to see. So we started working to try and uncover some of these variables by controlling them, and hopefully this can lead to uncovering the immunophenotype of Parkinson's disease by minimizing these background noise. So we worked with Abcam to provide them samples from our cohorts that have been sampled and collected and stored robustly to hopefully minimize some of this.
Wonderful. So then to take you through what the study design was, we decided to take a multi-omic discovery approach to looking at these neuroinflammatory biomarkers. And so what we really wanted to do was to utilize the fact that we could look at genomic and proteomic biomarkers on the same platform to see if there was a possibility that combining multiple analytes together might get us a little bit closer to finding these biomarkers. And so the approach was to use the FirePlex platform and to look at both microRNA and protein biomarkers in matched healthy and PD patient samples and in matched CSF and serum samples. And we also added a second variable of looking at whether the LRRK2 mutational status also affected any of the biomarkers that we identified. And so the reason for this particular approach was that, to the point that Nicole made earlier, it's potentially important to look at different matrices to see if we can find some of these biomarkers. So for example, CSF is not something that is very comfortable for patient collection, and so doing repeated measures is not an easy thing. So if we could find any biomarkers in the serum that might help point us to a neuroinflammation signature, that would certainly be something that we would like to look at as well. So in terms of the study breakdown, for the microRNA biomarker study, the question that we asked was, are there any microRNA biomarkers that can stratify healthy versus PD patient samples? And can we look at the serum and the CSF to see if we can identify any of these microRNAs? So we utilized a panel of 400 microRNAs that were specifically designed based on what was published to be found specifically in circulating biofluids. And this is particularly important because sometimes panels are designed looking at tissue-specific markers, which is wonderful, but if you can't detect those in a circulating biofluid, then they don't really apply to what you're trying to look at for a blood-based or CSF-based biomarker. For the protein biomarker studies, we wanted to really cast a very wide net, as I said, and we wanted to look at inflammatory cytokines and chemokines, and we really didn't want to restrict ourselves in any way to the analytes that we were looking at. And so the question was, can we identify an inflammatory protein signature in PD patient samples? And when we looked at the panels that we had currently in the FirePlex portfolio, or really what was available commercially in other companies, we saw that they weren't as comprehensive as we hoped. So we decided to actually take an assay development approach first, where we designed a specific 70-plex panel with guidance from the scientists at Michael J. Fox to try to include as many of these analytes in there as we could. And so a lot of the work that I'll take you through was this assay development work that our Immunoassay team did in Eugene, Oregon, to build and validate a 70-plex human inflammatory panel. And then once we had validated that this worked in multiplex in the sample types that we were interested in, we ran it with the samples that we had gotten from the Michael J. Fox collaboration. And so this is just a breakdown of how the study was set up. So for the immunoassay-based studies, we had a total of 70 patient samples split evenly between healthy and PD patients, and also split nearly evenly between male and female patients. These were matched CSF and serum samples, and of the PD patients, we included a subset with the that were positive for the LRRK2 mutation. For the microRNA profiling studies, we had a slightly smaller cohort of looking at approximately 38 patient samples, again split nearly evenly between healthy and PD and male and female. The one note that I want to make is that we knew going in that doing microRNA profiling from CSF was going to be a significant challenge. Doing genomics-based assays from CSF has proven to be significantly problematic for multiple assay platforms. And so what we found was that from the CSF samples that we had obtained, they were too dilute based on some pilot data to get robust microRNA signatures from it. And so what we decided to do was to pause that portion of the study and to go back and look and see if we could improve the sensitivity of detection in our assay before we ran it. So there won't be any CSF data for the microRNA studies portion of the talk. The other thing that I want to mention is that, as you can appreciate, we were asking a pretty big question from this patient study. So we were trying to address multiple variables of gender, healthy versus PD, and LRRK2 mutational status from this cohort. And so when I take you through the data, what you'll see is that we didn't find anything that was earth-shatteringly statistically significant that was different between healthy and PD. But that wasn't really the objective here. The objective was to look at a discovery approach and have it help us focus our studies so that we knew what subset of biomarkers we should be looking at, and then hopefully expand that to a larger cohort of patients to do some broad-scale screening studies. And hopefully the statistical power of those studies would allow us to identify clinically relevant biomarkers. Okay, so a quick intro into the assay workflows just to familiarize with you with how we do our assay detection. For the microRNA assays, we started directly with the serum or CSF, and we did a sample digest. We then label all of the released microRNAs with universal adapters, and this allows us to do a universal PCR amplification. The reason for doing a PCR amplification is that when you work with such limited sample inputs, in this case 20 microliters of serum and 40 microliters of CSF, the microRNAs that are present in very low abundance are difficult to detect. And so by doing the PCR amplification, we bring the signal up so that it falls within the linear range of the assay. But because it's a standard universal primer set, you reduce any of the amplification biases based on the sequence variations. So the microRNAs are captured onto the particles, we add on these adapters, and we do our PCR amplification. Within the PCR amplification, the product that we generate is biotinylated. We then add a reporter dye that is conjugated to streptavidin and then scan on a flow cytometer. And the readout that we get of the streptavidin conjugated to this red dye is how we do the analyte quantitation. So this is just a snapshot of typical data that we see with the FirePlex microRNA assays. So this platform has been validated for, as I said, a broad range of challenging sample types, and we expect to see robust signatures from these sample types. And we utilize the same approach when we were doing the studies that we're talking to you about here. For our standard throughput immunoassays, which are run in our 96-well plate format, the workflow is very similar to a multiplex ELISA, where you have capture antibodies that are conjugated to the FirePlex particles. We add these in solution with our sample. The analytes are captured onto the capture antibodies. We then add a detector antibody that is biotinylated, and again add the streptavidin conjugated to the reporter dye and do the assay detection. So all of the analysis by flow cytometry and the data analysis in the software is done exactly the same between the microRNA assays and the immunoassays. The really only difference is that for the microRNA assays, the input was either 20 microliters of serum or 40 microliters of CSF if we had run it. And for the immunoassays, it's 12.5 microliters of biofluid. So from less than 40 microliters of sample input, we were able to do both the microRNA and protein profiling studies. Now, as I mentioned, when we started to look at the portfolio of analytes we wanted to look at, we realized that we didn't actually have an assay, a panel that had all of these analytes in it. So we decided to combine Abcam's in-house antibody development technology with our ability to develop custom assays. And so for those of you who may be not familiar with Abcam, we have an in-house technology that allows us to develop recombinant monoclonal antibodies. And we do a lot of custom antibody development. And in particular, a lot of the relationship between Abcam and Michael J. Fox to date has been to develop these antibodies for difficult targets and targets that are of particular importance in Parkinson's disease. But more importantly for this talk, we also use that same antibody platform to do antibody pair development for all of the immunoassays that we have in-house. And those antibody pairs fuel both our simple step ELISA kits and matched antibody pair kits, as well as our multiplex immunoassays. So we decided to use that antibody technology, our custom assay development, and combine those together to make a custom 70-plex neuroinflammation panel. And so this is essentially a snapshot of what we did. So the FirePlex immunoassay scientists and the Michael J. Fox scientists came together to come up with a list of about 80 targets that we wanted to include in our panel. Then our team in Eugene, headed up by our principal scientist, Russ Nooner, set out to really do the panel development and validation. So they screened all of these antibody pairs first in small panels, and then finally in the full 70-plex panel to validate that it could be run in multiplex without any interference or crosstalk or anything like that. Once the pair screening and panel development was complete, we did the validation in the biofluid sample types. I'll show you some of that data in a moment. And then when they were ready to go, we ran the samples that we had obtained from the foundation. So this actually ended up being a multi-site global effort that we took on. So as I mentioned, Sandy is our alliance manager, and she was the person who really helped make sure that this project was moving along and we were hitting all of our milestones. And then our custom immunoassay development team in Eugene really did a tremendous job of developing the 70-plex panel in about 6 months. And for anyone who's been involved in multiplex panel development, that is an outstanding accomplishment. It takes a really long time to do pair screening, but we were really excited that we could do that in short turnaround for these studies. We also had our R&D microRNA team who developed the 400-plex panel, and all of the antibodies that were used were developed in our team by our teams either in Hangzhou or Cambridge, UK. So once all the assays were built, they were sent to the FirePlex service lab in Boston, that is headed up by one of our very talented colleagues, Mike Tackett. And his team then ran all of the samples that were provided by Michael J. Fox on the microRNA and protein panels. And of course, there was a huge amount of input from the team at Michael J. Fox as well. So Nicole and Tarina and Anna really helped inform a lot of the panel design. And then we had studies from Italy and Illinois that supported the biofluid sample types that we used. So just to show you a little bit of the performance data for the 70-plex panel, when we developed this, what we wanted to ensure was that we were seeing sensitive detection of all of the analytes, we weren't seeing crosstalk between the antibody pairs, and that we were retaining the dynamic range that we needed because we knew that some of these analytes were going to be present at fairly low concentrations. And so obviously this is a very busy graph, but what it's just meant to show you is that for all of the analytes in the 70-plex panel, we're retaining about 5 logs of dynamic range. So we expected to be able to sensitively detect all of the analytes, and we're not seeing any issues with antibody cross-reactivity or anything like that. Obviously, these are just standard curves, so this is a little bit of a vacuum, but what's going to be more important is to look at these in biological sample types. And I apologize that this legend is a little bit small, but we validated, I will read it to you. I can barely read it, so I can imagine it's hard for you to read. We validated it in all of the sample types that we routinely screen for our FirePlex platform. So this was plasma that was either heparinized or not, serum, urine, CSF, bronchial lavage fluid, and cell culture supernatants. And so the graph that you're seeing, each of the colors represents the different sample types. And what you can see is that we detect the majority of the analytes in all of these sample types. Now, you wouldn't necessarily expect to detect everything in every sample type because it may not be present, but what we were really looking for was robust detection of the targets we expected to detect, and we certainly saw that. So once we saw that and we knew we had validated it with our in-house samples, we felt confident to then move ahead and run the Michael J. Fox Foundation samples with it. All right, so finally, getting into the results, I will begin by taking you through the immunoassay data first. So this is a snapshot of the data output that we saw for the serum immunoassay profiling studies. And for the 70 samples that we looked at and the 70 analytes in each of these samples, we detected 53 of those analytes above the limit of detection. Of the 17 that we could not detect, 6 of them fell outside the upper range of the standard curve, meaning that they were present in too high a concentration, and the remainder fell below the limit of detection. And so that really points to the fact that when you do such a large plex study, sometimes you then have to adjust the sample dilutions and repeat. So that's something that certainly we're going to look at. So we wanted to start by looking at some of the targets that Nicole mentioned earlier and those that we had already sensed that might be involved in PD. And for the majority of them, we expected that they would be upregulated in PD based on the neuroinflammatory component. So when we look at the results, it's a little bit of a mixed bag. What we saw was that for IL-1 beta and IL-2, those were trending in the right direction. However, they were all detected fairly below the limit of quantitation. And so for these would benefit from being repeated at a lower sample dilution. For IL-6 and IL-10, we could detect them above the limit of quantitation. However, we didn't see statistical significance in the data, even though they were trending in the right direction of being upregulated. Again, that suggests that we just need a slightly larger cohort when we go back to repeat these studies. And then finally, for interferon gamma and TNF alpha, we could detect them above the limit of quantitation, but there was no statistical significance in these studies, so really would benefit from a repeat with a larger patient cohort. So this is just a snapshot of some of the analytes that we identified. So the table on the left is looking at PD versus healthy patient samples. And what you can see is that there is a series of targets that are statistically significant in being differentially regulated in PD. But none of these were ones that were necessarily published in the literature previously. So this is something that we would have to certainly investigate the importance of some of these analytes to see what we can identify. Interestingly, when we look at the LRRK2 positive versus negative patient samples, we do see some targets that are also differentially regulated. So again, this would be something that would be really interesting to further investigate. We also did look at whether there was a gender-specific component in these samples. So we looked at male versus female samples, again, PD versus healthy. And we do see a couple of targets, although the hemoglobin and the myoglobin are likely some indication of hemolysis may be happening. But in general, there are some targets in there that may be worthwhile to investigate. Looking at the CSF data, we again looked at the number of targets that could be detected above the limit of detection. And of the 70 samples that we looked at and of the 70 analytes in that panel, we could detect 42 of the analytes above the limit of detection. Of those, of the 28 we couldn't detect, 3 fell outside of the upper range of quantitation on the standard curve, and the rest fell below. So the case of CSF was, as we had seen with the microRNA, the samples are just fairly dilute. And so we would need to go back and repeat this, particularly for the targets that we couldn't detect at a lower sample dilution to hopefully bump the signal for all of those back into the linear range of the assay. And again, we took the approach of looking at what was already known in the literature to see if that could help inform some of the data that we were looking at. And what we found was similarly to what we had seen with the serum data, the IL-1 beta, interferon gamma, and TNF alpha are not present in the linear range of the assay. We couldn't quantify them, and so this would really bear repeating at that lower sample dilution that I mentioned. And then for the others, so IL-6 was present and detectable. However, we didn't see any statistical significance when we looked at the data, although I will show you some data on the LRRK2 patients for IL-6. That's a little bit interesting. And then the PGE2 and osteopontin weren't included in this initial panel design, so we can certainly go back and include those in future panel designs as well. So looking at the PD versus healthy patients, we didn't identify any targets in CSF that were statistically significant in being differentially regulated. But there are some really interesting targets that jump out in the LRRK2 positive patient samples, particularly the IL-6, as well as some of these other interleukins. And so given that there was only 10 patient samples that we looked at between the LRRK2 positive and negative patients, this would be something that would be really interesting to further investigate with these larger patient cohorts. So I'm excited to see what may come of that. And it is encouraging to see that it is upregulated in the LRRK2 positive patient samples for IL-6, which is what we expected. And then looking at the CSF data, male versus female samples, we do identify these targets of IL-8 and liver fat B. So I'm not sure what the clinical significance of this would be and whether potentially in CSF there is a potential for a gender-specific signature, but this would be something that we would certainly need to investigate further. And again, this was really an objective of trying to look at all of these analytes to see if we could refine that panel down to a smaller set of targets so that we could do these larger scale patient studies and get some usable data from it. And then very quickly looking at the microRNA data, as I mentioned, we solely focused on the serum for these studies. So for the 36 samples that we looked at, 110 out of the 405 microRNAs that were present in the samples could be detected at a mean fluorescence intensity above 1. And so what you're seeing in this heat map is sort of the spread of microRNA profiles. And so for the majority of them, we're seeing low fold changes. This is a log 10 scale, but then there are some targets that we do see pretty significant fold change differences in. Now, interesting for the microRNA data, while we didn't see any microRNAs that jumped out as being differentially regulated between the LRRK2 positive and negative samples, we did see a couple of microRNAs that were statistically significant between PD versus healthy patients. And what's interesting to note is that miR16 is a neurospecific microRNA that's really robustly detected in neuronal sample types. And so it's interesting to see its enrichment in serum in the PD patient samples, potentially suggesting that there may be some microRNAs that are entering the blood in response to this PD. Again, absolutely bears further investigating. And then looking at the male versus female samples, again, we see some microRNA signatures that are differentially regulated. This was a slightly smaller study, and so I do think that if we expanded the patient cohort size, there would be some additional microRNAs in there that we may identify as being differentially regulated. And what's particularly interesting to me from these studies is that I understand from Nicole that there are collaborators within the Fox Foundation network who are running RNA-seq studies. And so it'd be interesting to use the data from the RNA-seq to inform a panel design that could be more focused on these PD-specific microRNAs and then to repeat these studies again. Okay, so the last couple of slides, we just wanted to really touch on what we have learned and where we think that the future directions, not just of this particular study, but of neuroinflammation and PD will go. And then, of course, some closing thoughts.
So just to recap the landscape of neuroinflammation and PD, we have improved tools, patient cohorts, and data sets that strongly implicate a role for neuroinflammation in Parkinson's disease. Some examples of this are through GWAS studies. There are variations in antigen-presenting genes that confer a slightly increased risk for late-onset Parkinson's. Preclinical models of Parkinson's have an inflammatory component present that contributes to nigral neuron degeneration, and interfering in that process can alter pathology in these models. Epidemiological studies have shown that chronic NSAID regimens early in life can lower the risk for Parkinson's later in life. Mutations in LRRK2, which is a genetic risk factor for Parkinson's, have been linked to inflammation through multiple studies and mechanisms. There are still outstanding questions, such as what is the role of inflammation in the gut and how may that contribute to Parkinson's pathology? What's the impact of misfolded proteins on initiating inflammation? A hallmark of Parkinson's disease is misfolded aggregated alpha-synuclein. Is that possibly triggering this process? What is the role of neuroinflammation in other neurodegenerative diseases, and how can efforts be synergized between different foundations, different labs, different groups to kind of learn from each other and move the needle forward in multiple disease states? And also, what is the role of inflammation that should be considered in the development of disease-modifying treatments for Parkinson's? So can we develop treatments to target neuroinflammation in Parkinson's to reduce pathology? Can we develop imaging tracers and biofluid biomarkers for patient stratification, measures of disease progression, or maybe diagnosis? And can we develop improved assays and tools for PD biomarker discovery and screening that can help us with these efforts? And especially those last two are highlighted because those are really what our focus was in this effort.
And so really the last slide that we wanted to leave you with is this sort of snapshot into the biomarkers of neurodegeneration. So this is really not a comprehensive image, but it's just really meant to show you that there's so much research that has gone into trying to inform these circulating biomarkers in neurodegenerative diseases. And obviously the projects like the ones that we were lucky enough to participate in with Michael J. Fox are aiming to help advance that. We typically have more questions than we do answers, but it gives us a really exciting starting point to try and tackle some of these problems. And so with that, I want to thank all of the people on these slides who were involved. So first of all, to Nicole and the rest of the Michael J. Fox colleagues, again, this has been a labor of love for 2 years of lots of discussions about project design, project scoping, panel design, analyte, sample types. It's been a great collaborative effort, and I'm so appreciative. And to all of our collaborators within the Michael J. Fox Foundation and at Abcam, particularly Sandy, who manages the Abcam-Michael J. Fox relationship, and then, of course, all of the other assay development scientists who were involved in the panel and project design. And so thank you all for being here. And of course, if you have any questions, we're happy to help answer them here, or you can come visit us at our booth. Thank you. Are there any questions? Yes.
Did you look at correlation between the CSF and serum samples?
That's an excellent question. So the question was whether we looked at the correlation between CSF and serum samples. We did not. That's something that we can certainly go back and look at. Yeah. Because that, to your point, one of the questions that we asked is, do you see an enrichment in some of these biomarkers from either CSF into serum or vice versa in disease progression? Yeah. Yes.
So you've done a lot of work with protein and RNA. How about lipids? For example, how about prostaglandins? It's so well established and helps to definitely in Parkinson. And this field is so unexplored. Maybe we will get some better biomarkers. So I'm very interested in having these tools to analyze prostaglandin profiles.
Absolutely. So the question was whether, in addition to the genomic and proteomic biomarkers, we've also looked at lipids that are obviously implicated in PD. So at the moment, we don't have a way of looking at prostaglandins or other lipids on the FirePlex platform. However, within our portfolio at Abcam, we do have assays that look at that. And so one of the future directions that we are exploring is whether we can bring in other assay platforms to inform this, or if we can do some finagling of assay development to somehow look at lipids on the particles. So it's definitely part of the things that we'd like to investigate, certainly, yes. I think there was another question maybe in the back. Or I just answered it. Yes.
So I know that you've developed this for human studies, but there's so many amazing transgenic mouse models that can be evaluated.
Absolutely.
Are you planning to make a multiplex system that can evaluate differences in treated and untreated mice?
That's an excellent question as well. So the question was, all of the data that we took you through and all of the panel development was done for human samples and human analytes. And so the question was whether we plan to do the same for mouse samples. So actually the mouse 70-plex cytokine discovery panel just completed assay development last week. One of our assay development scientists is in the back. Actually, she was telling me about it yesterday. So that will be probably ready to go in about a month's time. And then we would be able to apply it to those studies, certainly. Yeah. Yes.
I have a question about your agenda.
Please. Yes, of course.
Because you talk about, the dynamic range is really impressive. You showed that in your figure. But it says that it's relative to other platforms.
So that's a good question. The question is related to something that's in the FirePlex literature, looking at the dynamic range of the assays. So what we did is the dynamic range, for example, in the standard curves that I showed you, is just the performance within our own assay. What we did is we looked at the published sensitivities and published dynamic ranges for other bead-based assays, comparing them to FirePlex. And so what you're looking at is a data sheet versus data sheet comparison, and essentially looking at the number of targets that we can either more sensitively detect or have a broader dynamic range in our portfolio versus on the other bead-based providers. And I can take you through more of that if you'd like. Yeah.
Does the multiplexing increase the sensitivity for any individual one?
That's an excellent question. So the question was whether the multiplexing decreases the sensitivity. So as a part of the 70-plex panel development, that was one of the first questions that we asked, because we knew we were working with fairly dilute samples, or we needed them to be as sensitive as possible. So the assay development team actually started by building 8 smaller plex panels that they then tested the performance of and started to piece together in these modular entities. And one of the criteria that we had for panel development was that we not lose sensitivity of detection in 70-plex compared to like a 10-plex, because we could just simply run more panels if we needed to. And so based on the standard curve, what you see is that the limit of detection and lower limit of quantitation is almost identical between the 17-plex panel that we had and the 70-plex panel. So we don't really see a loss in performance there. Please.
So based on the results that you got,
Yeah.
So based on that kind of first pass result, did you ever subdivide panels? So that you then have your high dilution panel or your low?
Right. So the question is related to the analytes that for serum and CSF fell outside of the upper range of the standard curve, which honestly, I don't think many of us were expecting. Do we plan on rebuilding the panels and splitting them out? And absolutely yes. So one of the other panel development criteria that we employed is a 70-plex panel is only useful if you can do a single dilution and detect everything. And so the strict criteria we had was that everything had to be dilution matched, and these were all run at 1:4. What we knew, though, was that we'd run these studies and then we would have to dilution adjust. So the goal is to go back and split that panel out for the targets that fall outside the upper limit of quantitation, split them out and do higher sample dilutions. And for everything else that fell below the limit of quantitation, split those also out and then do lower dilutions for those. But the good news is that you can still start with one input and do the serial dilution, so you're still not going to use way more sample than you would. So yes, that's going to be in phase 2 of these studies. Any other questions? Awesome. Well, thank you all again for being here, and thank you, Nicole.